the results we expect are similar. We expect the side to culture some bacteria, so resting bacteria from our same body. And in the clean side, we expect those bacteria to disappear or significantly decrease. All right, now let's go to the exercise 2. So, what we're going to learn with exercise 1 is that bacteria are present everywhere. We're carrying them, and what we're going to do on exercise 2 is how to avoid the presence of this bacteria in our experiment. Now, to truly understand a microorganism, we need to study that microorganism separately from the rest. So we can properly identify it, we can properly test the metabolic tests that we want to do, and we want to learn how to keep every single media that we are going to work as clean as possible to avoid contamination and mistaking one microorganism for the other. In order to do that, in microbiology, we use a technique called aseptic technique. So, bolded words in this class are bolded for a reason, and so my advice is that you pay close attention to them and try to learn them. So, aseptic technique is a collection of procedures and rules that we use to minimize the chance of contamination. It's a very simple technique that I will demonstrate later on, in which we use an inoculating loop, a Bunsen burner, to maintain what we call an aseptic or contamination-free environment. So we'll go into more detail with this when I do the live demo. So we are going to learn how to perform 2 basic bacterial transfers using aseptic technique. The first transfer is a slant to a slant transfer. So transferring from one bacterial slant to another bacterial slant. And then what we call a slant to plate transfer. So transferring from these to a petri dish. Bacterial slants are used mostly for the storage of bacteria. They take less space, a little bit more easy to store. And then the plates are a better way to culture them and grow your bacteria. So I'm going to go into the details of this when I do the live demo. And now let's go to the other technique that we're going to learn that connects a little bit with studying those microorganisms. As I was telling you, to truly understand a microorganism, we need to isolate those. Now, because microorganisms, with a very few exceptions, are very difficult to distinguish with the naked eye, we need to isolate them. And to make sure that we are working with only one species, strain, or group of microorganisms. So to do that with bacteria, we use a technique that is called streaking for isolation that will help us isolate and select genetically identical strains or types of microorganisms. Now, this technique is based on the concept of diluting or thinning out the number of cells that we apply into a petri dish until we obtain individual separate cells that later grow onto a colony. I guarantee the concept of colony is going to be at least in one test, so learn it. A colony is a group of cells which arise from a single cell. The most important part to obtain full credit on this question is which arise from a single cell. What this guarantees us is that the genetic makeup, assuming there was non-mutation in the reproduction of these little dot right here, is identical. And that's the way that we isolate those bacteria. The procedure itself I will explain in a better detail during the day. Alright, so if you more or less can see from this image, what we are doing is we're culturing a large number of bacteria on one side, and then we are decreasing that number of bacteria as we spread it in the active. So, what we're going to do now, guys, is you're going to come up to the front, where I will do live demos on these techniques that I just talked about, and then you guys are going to perform it later. Just a heads up, we're going to record this for internal use purposes, but they're going to record me. There might be some of you that are in the frame, so I just want to let you know that that's going to happen. All right.